For the longest time, we pictured the brain as this untouchable fortress, totally sealed off from the rest of the body's chaotic immune system, a kind of protected, privileged kingdom. Well, turns out we were wrong. The brain has its own private security force, its own secret agents, and figuring them out is rewriting literally everything we know about how our brain works, how it ages, and how it breaks down.
And that right there is the central paradox we're going to dig into. Because the story of the brain's immune system, it isn't some simple tale of good guys versus bad guys. Nope, it's about a guardian that, under just the wrong circumstances, can become a saboteur.
So this idea had a pretty cool name, immune privilege. The thinking was, the brain is just too important, way too delicate, to let the body's heavy-handed immune cells come storming in. There's this super complex barrier, the blood-brain barrier, and we thought it kept them out for good.
But here's the thing. The brain isn't defenseless. It doesn't need to borrow guards from the rest of the body because it has its own. A super specialized, dedicated team of cells that are born with the brain, live with the brain, and die with the brain. And their one job? To protect it from the inside out.
And these cells, well, they have a name, microglia. They are the main characters of our story today, the secret agents at the very heart of brain health and, as we'll see, brain disease.
So to put it simply, microglia are the brain's personal immune system. They're the first line of defense, always on patrol, acting as guardians, housekeepers, and emergency first responders for our entire central nervous system.
Just finding these cells was a detective story that took 100 years. It all started back in the 1800s with this vague idea of a nervous glue holding our neurons in place. Then, the legendary neuroscientist Gajal spotted this mysterious third element that wasn't a neuron or any other cell they knew. But the big breakthrough came in 1919 when his student, Pio del Rio Ortega, finally managed to isolate these strange, spidery cells and gave them the name microglia. And what's just amazing is that he guessed they came from outside the brain, an idea so out there it took almost another 100 years, all the way to 2010, for modern genetics to prove he was absolutely right. They actually move into the brain when we're just tiny embryos.
And just look at these incredible drawings from Rio Ortega's original 1919 paper. They show exactly what he saw. Over on the left, in panel A, that's a healthy, resting microglia, with all its delicate branches just constantly checking out its surroundings. But then look at B and C. After an infection or an injury, they completely transform, pulling in their branches and becoming these little blob-like movers and shakers, ready to jump into action. He saw over a century ago that these weren't just static little specks, they were dynamic living responders.
So what's a day in the life of a microglia like? Well, their to-do list is huge. They're in constant surveillance mode, their little arms are always reaching out, kind of tasting the environment. They're the brain's cleanup crew, gobbling up all the cellular trash and the remains of dead neurons. And, this is fascinating, they're also sculptors. They prune away weak or unused connections between neurons, which actually makes our brain circuits faster and more efficient.
But, and this is a huge but, there's a twist. This perfect little security system has a dark side. The very same instincts that make microglia such amazing protectors can also turn them into agents of destruction. This really captures their Jekyll and Hyde personality perfectly. In a healthy brain, they are total heroes. They clear out those toxic protein clumps we hear about in Alzheimer's, and they release signals to calm everything down. But if they get overactivated, it's like a switch flips. They start pumping out toxic, inflammatory molecules that basically create a hostile environment in the brain. And in that frenzy, they can start destroying perfectly healthy neurons as collateral damage.
This quote from a team of researchers really hits the nail on the head. An immune system that's out of control just creates this vicious cycle. Inflammation triggers more inflammation, which leads to this state of chronic, simmering damage that can be absolutely devastating for the brain.
And look, this isn't some rare, theoretical problem. This double-edged sword of the microglia is now understood to be a key player in a huge range of the most challenging brain disorders that we're facing today.
I mean, the list is just staggering. It connects microglia to so many conditions we never thought were related. We see their fingerprints all over classic neurodegenerative diseases like Alzheimer's and Parkinson's, but also in autoimmune conditions like MS. Their influence even extends to brain tumors and, maybe most surprisingly of all, to major depression. It shows these tiny cells are at the scene of the crime across a huge spectrum of brain disorders.
So how does this actually work? Well, in Alzheimer's, it's not a big sudden explosion. It's more of a slow burn. The microglia respond to that buildup of amyloid plaques, but the response is kind of weak and inefficient. So instead of cleaning it all up, they just get stuck in this state of low-level chronic irritation. This simmering inflammation just goes on for decades, slowly cooking the brain and contributing to the death of neurons.
And we see a really similar pattern in multiple sclerosis. After an immune attack creates a lesion in the brain, the microglia at the edges of that lesion can stay active permanently. They just don't switch off. This creates what researchers call smoldering damage, a continuous low-level attack that prevents healing and helps the disease get worse over time.
And what about depression? This is where it gets really interesting. Recent breakthroughs have shown it's not just a matter of brain chemistry. By actually analyzing brain tissue from people who had depression, scientists found altered gene activity specifically within their microglia. This is powerful evidence that inflammation in the brain plays a direct role, really reinforcing that depression has a clear, measurable biological basis.
So is there a tipping point? When does our brain become most vulnerable? Well, research is pointing to this one critical age, around 57. This seems to be when the brain's protective barrier, that blood-brain barrier we talked about, can start to get leaky. And this breach doesn't just let harmful stuff in from the body, it creates a constant state of alarm that keeps our microglia triggered, stuck in that chronically inflamed, destructive state that can lead to diseases like Alzheimer's.
Okay, so rogue microglia are clearly a huge problem. But here's the flip side. This new understanding opens up an absolutely incredible new opportunity. What if, instead of just fighting the brain's immune system, we could learn to control it? This, right here, is the next frontier in neuroscience.
The old approach was basically to just use blunt anti-inflammatory drugs to shut everything down, but the new, much smarter goal is modulation. The idea is to tame the harmful, destructive microglia while at the same time boosting their helpful protective functions, like cleaning up all that debris. It's about rebalancing the system, not just nuking it.
And scientists are already developing these mind-blowing tools to do just that. For example, they're finding ways to boost a protein on the microglia's surface, called TREM2, to make them better at clearing plaques in Alzheimer's. For MS, they're targeting the internal power source of overactive microglia to selectively shut them down. They're even using special antibodies to basically reboot an aging immune system so it can fight brain tumors better.
But maybe the most futuristic strategy is this one. Using other immune cells, called T cells, as programmable drones. Here's how it works. Scientists engineer the T cells with a molecular GPS that tells them to go only to the brain. Once they cross into the brain, a second sensor activates them only when they find their specific target, like a tumor cell. Then, and only then, do they release their therapeutic cargo. It's an unbelievably precise, targeted delivery system.
So when you step back and look at the big picture, we've gone from not even knowing these cells existed to being on the verge of actually reprogramming them. The brain's secret agent, once a total mystery, is now becoming an ally that we can potentially direct. And that shift could fundamentally change how we treat everything from Alzheimer's to depression, opening up a whole new chapter in the story of brain health.